This method is significant because patient-derived organoid models are a powerful and rapid tool to study pancreatic cancer, as they reflect the genetic and phenotypic heterogeneity of this disease.
The main advantage of this technique is that organoids can be isolated with a high success rate from a limited amount of cancer, and they can be expanded rapidly in vitro for downstream analysis.
Studying organoids' response to cytotoxic drugs may help us understand why tumors become resistant to therapy and to define more effective treatment strategies.
To begin this procedure, retrieve the prepared 12-well plate from the incubator. With the sterile cell lifter, gently lift the basement membrane extract dome such that it is floating in the complete media while making sure to avoid scraping the bottom of the well. Using a P1000 pipette, carefully transfer the BME dome and the media to a 15 milliliter conical tube. Repeat this step for every organoid containing well. Gently wash each well that was harvested with 1 milliliter of cold basil media and transfer this wash to the tube containing the organoids. Thoroughly mix the solution and organoids with a P1000 pipette and spin down at 200 times G for 5 minutes. A layer of BME containing organoids in suspension and an organoid pellet will appear at the bottom of the tube. Carefully remove the supernatant, making sure to avoid any loss of the BME and organoid layer. Place the tube on ice. Add 10 milliliters of ice-cold cell recovery.